What's up, y'all? I realized after posting the video that I never went back in transcription and translation to work on that little chart with you guys that helped us tell the difference between these processes. It is important enough that I decided to do a little short add-on video just to make up for that content. So let's share this through and get this thing done. We did the previous parts of this. We did skip this and say we were going to come back, and then we never did. So here we are. It's time to catch up. Transcription, translation, and replication. I'm going to give you brief descriptions again, just to recap in case you're not watching this right after. Replication is when we copy our DNA. This happens in the S part of interphase. We're making extra copies of our DNA specifically so that a new cell gets full sets of information. Transcription is using DNA to make an mRNA copy of genetic information, a disposable copy of information that we can then take and use to build something. Translation is when we take that mRNA copy that we made in transcription and we bring it to a ribosome and physically build the protein by looking at the code in the mRNA and calling in amino acids to get put into the correct orders to make our protein. So let's see what we've got here. Creates an identical copy of DNA. This is going to be replication. Replication is the only one where we're just making a copy of DNA. In transcription, we're making a copy, but we're making an mRNA copy, not just more DNA. DNA strands separate from each other. This is happening in replication when we have to open things up to be able to get at the code inside and build our new halves. It's also happening in transcription because we have to open up the DNA so that we can get at the code to make our mRNA copy. At the end of transcription, we pull out our mRNA copy and just close the DNA again, but we still had to open up the DNA to get at that code. The DNA is not involved directly in translation at all, so that one does not make our list for this second box. Amino acids being linked together in a polypeptide. Polypeptide is a science code word for protein. So when we're actually building the protein would be that third and final step, translation, where we assemble those together based off of the genetic info. Now this one says takes place in the cytoplasm or takes place in the nucleus. Think logically. If I'm in the cytoplasm, I'm nowhere near the DNA. I know this because DNA has a house. It's called a nucleus. And the DNA stays locked up nice and safe inside of there, which means the only part of these that could happen in the cytoplasm would be if I did not need the DNA. Well, as I mentioned a moment ago, the only one where I don't need the DNA is actually translation, because that's the one where we're building the protein. If you built this protein inside of the nucleus instead of in the cytoplasm, you'd never get the protein out. It'd be too big to fit through the door, and then it's kind of a waste. What did you make this protein for? We can't even take it to where it's supposed to go. Now, the other two, replication and transcription, those are both happening in the nucleus because they both have to have access to the DNA, and the DNA does not leave the nucleus unless it has to during cell division. Requires tRNA and mRNA. tRNA is transfer RNA, the one bringing the amino acids in. mRNA is the copy of the genetic code that we're using. The only one that uses them both is translation in the building of that protein. Uses information from a gene to produce a specific protein. In my opinion, these are all kind of related to each other in this case. But if they're saying using the information from a gene to produce the protein, The only one actually producing anything is translation. Two left uses ATC and G nucleotides. If we rearrange those, that makes up GCAT. GCAT is used in replication because it's DNA related. And the GCAT bases are part of the DNA that gets used in transcription, but we actually build using GCAU because it's mRNA in transcription. So using GCAW bases instead of GCAT would be transcription and translation. Y'all, I had to come back and finish this chart for you, even though it's a little mutant video that's added on. Had to do it because I love you guys. I can't leave you hanging like that. I told you I'd come back to it. I came back to it. Hopefully everything's good. I will catch you in the next one. We will see you next time.